All right, another thing that is going to affect this overall issue is the idea of multiple alleles. So before we've just been working with two, now we can see that the blood groups are actually made up of three separate alleles. So there's capital I A, there's capital I B, and then there's little I. As you can probably guess from the lettering scheme here, little I is recessive, and in this case actually refers to a form of the gene that is non-functional. So you might ask the question, well, what do the functional enzymes actually do? So in this case, the A form adds a particular sugar, and there is a mutated version of it that adds a different sugar. It's actually add sugars onto a growing sugar chain that is going to be expressed on the outside of our cells. So here we're going to show it in terms of red blood cells, but it's actually on the surface of all of our cells. So since the A form is capable of adding this triangle-shaped sugar, of course, carbohydrates aren't triangles or circles, but just as a representation, this cell here, which could be genotype big A or capital IA, homozygote or heterozygote, it will express this triangular sugar on its surface, and this is what we know as blood type A. In the case of B, it's going to add this different sugar, and so either the homozygote or heterozygote of this will produce our blood type B. In the case of having heterozygote for A and B, as I said before, these are codominant, so consequently each cell will have both versions. So have both the triangular version and the circular version here. That produces a blood group that we now are going to refer to as AB because it's a combination of both of these. Little I, so the homozygote recessive in this case isn't going to express any of those. And that leads us to blood sugar O. And this actually helps explain why O is a universal donor. If our immune system is recognizing this form and it is now given this B form, it'll recognize the B form as foreign, coming from the outside, and it'll attack it and it'll destroy it. If you are type A and you get type AB, the same thing is true. You will recognize the presence of the B antigen in this case as foreign, and you will attack and destroy it. If you get type O, you can accept that because the enzyme that is adding the A is actually not 100% efficient. So we have the precursor sugar chain that is present in O present on all of our cells. So O can be accepted by anybody because it doesn't have any added markers. If you are type AB blood, you can actually receive blood from anybody. So here you can receive A because you don't see that as foreign. You can receive B because you don't see that as foreign. You can receive O because you don't see that as foreign. So if you're on the injury end, AB is your best case scenario because you can get blood from anybody. If you're looking for a donor, you're looking for type O because you can contribute blood to anybody. So this is what happens when we start getting multiple alleles. And here we can see that not only do we have multiple alleles, we have three, but we also have an example of codominance occurring here. Now, as I said before, genes can be different or have different phenotypic effects. This is the name for this is pleiotropy. So if we were to give the example of cystic fibrosis, which I mentioned before, it is a defective protein. It can be expressed in the lungs and has one phenotype there. In the gut, it has a different phenotype. In the skin, it has a different phenotype, right? So depending on where it is and what its job is in that location is going to change how that phenotype is expressed, right? So most genes have multiple effects. Now, when you start extending various traits that are regulated by two or more genes, we need to start thinking about it in a slightly different way. So I would say most of our traits are actually determined by multiple genes. And this is why, although there may be a genetic background that predisposes you to a number of conditions, we can't really figure out how that works yet, right? So we know that there are inheritable traits that leads you to heart disease, but we don't really understand what all those are because it is controlled by multiple genes working together. So let's give one particular example here where we have a gene at one locus is actually going to alter the phenotypic expression of a gene at a second locus. So one gene product is actually controlling another. This is something that we call epistasis. An example of this would be in laboratory retrievers. So, as you may know, you can have black laboratory retrievers, you can have brown laboratory retrievers, or also known as chocolate, and you can also have yellow laboratory retrievers. This is based on the fact that we have a two-gene scenario. So one gene labeled as B determines the pigment color. So it's either going to be black or brown. There is a separate gene, gene E, and this determines whether or not pigment will be deposited in hair. So in this case, if you have a lab with little e, you have no pigment deposited into the hair, so it doesn't matter if you're big B, big B, big B, little b, or little b, little b. You will be yellow regardless, and you are not depositing any pigment. In this case over here, we kind of see the normal phenotype of looking at just the B locus. So here we have big B, little, or big B is black, big B, little b is black, big B, little b over here again is black, so a 3-to-1 ratio, so little b is brown. Okay. But we also have these other variations too. So this combination of these two genes together essentially deviates from that 9-to-3 to 3-to-1 ratio, kind of collapsing the final 3 and 1 into a single group of 4. So now we have a 9-to-3-to-4 ratio instead of what we would normally expect from a two-gene scenario of a 9-to-3 to 3-to-1 ratio. And this is because this E gene is essentially negating what the genotype for the B gene is. All right, so we talked about pleiotropy, we talked about epistasis, but what I want you to recognize that there are a whole lot of genes that actually fall along what we know call quantitative characters. So these are things that exist along a spectrum, and this because it's a polygenic scenario. So you actually have many genes working together, and the additive effect of these genes is leading to the phenotypes that we see. This is the case for skin coloration. So here we're going to represent a simplified scenario. Here we are going to have 3 genes. Each one of these 3 genes is adding more color to the skin. In reality, this is actually controlled by 16 genes. Okay, so we're making it real simple here. So in this case, you have one copy of that gene from mom, one copy of that gene from dad. And you can have essentially all these different combinations. So you can have all 6 being lighter in color, or you can have a single dark skin allele, or you might have 2 dark skin alleles, or you might have 3 dark skin alleles, or 4 dark skin alleles. You can see how this overarching grid is producing a whole palette, right? And this is all dependent on the number of these factors that are coming into play. As I said, this is actually not just 3 genes, this is actually a good 16 different genes, and some of those genes are interacting in a pleiotropic type way. So it's coming back to what we, or I'm sorry, in an epistasis type of way. So it comes back to what we see here. So in reality, this is way more complicated than even this complicated picture is demonstrating. Now, if all of this wasn't enough, sometimes nature is making that determining scenario. So recognize that our bodies are designed to interact with the environment around it, and that can produce differences based upon the environment we're in. So one example of this is these, I believe they're called hydrena bushes, but they produce these colorful flowers, and the coloration of the flower is actually dependent on the pH of the soil that it's growing in. So you can take this pink plant, plant it in a new scenario, and it will become a blue flower plant. So this is one example where the pH of the soil that it's growing in is making an impact. If you look at other factors, you can see things showing up there as well, such as hair coloration. So you may have seen examples before of animals that live in temperate environments that can change colorization depending on season. So during summer months or warmer months, it has colors that will help it camouflage. Become winter, it will replace its coat with a white coat, allowing it to hide better in snow. Both of these are examples of the environment impacting how our genes play out. So, again, concept 14.3, inheritance patterns are awfully more complex than predicted by simple Mendelian genetics. Gave you the example of incomplete dominant, or before we gave you an idea of complete dominance, but here we gave you an idea of incomplete dominance, right? So instead of um, just having the functional gene giving you the purple coloration, here you have red or white, but when you combine the two, a single copy of the gene isn't enough to make it red, but it is enough to make it pink, right? So we get different ratios. In this case, the ratio is the same as the genotype. So we can extend these patterns to deal with the scenario of when we have non-completely dominant genes or non-completely recessive. We might also need to adjust this to multiple alleles or to multiple phenotypes from a single gene. So we gave you an example here of blood type. Here we have multiple alleles, and 2 of these happen to be codominant. So this combination of 3 actually gives us 4 different phenotypes. We could have A, B, or O. We gave you the example of epistasis, where we have one gene controlling another.